of Coimbra, Portugal. He did his postdoctoral work at INET, Portugal, and MIT, USA in the areas of stem cells and nanotechnologies. He established his research group in 2008 in the University of Coimbra. Then he is the director of the Advanced Therapies Research Group, CNC coordinator of the MIT Portugal program, and the founder of the biotech companies Matera and Curamed. Since 2018, he became the associate editor of Biomaterial Science Journal. In 2012, he was awarded the prestigious European Research Council Award, and in 2016, a prestigious ERA chair position in the area of aging at the University of Coimbra. Since 2020, he is the coordinator of the Collaboration for Aging, a collaborative laboratory that integrates companies, health and social providers in the area of aging. The floor is yours, Lino.
Thank you so much, Mehmet, for the kind words. So I'm going to speak about stem cell-based models and advanced formulations to study and treat aging. So I'm affiliated with the Faculty of Medicine at the University of Coimbra in Portugal. So I'm located in this corner of the world. So Portugal is in the west part, the most west part of Europe, close to the Atlantic Sea. So I'm located in the center region of Portugal. So this is my group. And the University of Coimbra, for those that are not aware, it's one of the oldest universities in the world. So it's in the top 10, in fact, the top 10 universities, world universities. So Bologna is the first one, and I think Coimbra is located in the 9th place. It's a very beautiful university, as you can see here. It's since 2013, it belongs to the World Heritage, and so I invite you, if you have the possibility to travel to Europe for a conference and so on, to visit us. So this is the institute that I belong. So it's integrated at the University of Coimbra, and these are some of indicators in the last years. So my group is in the border at the interface between aging and bioengineering. So at the stem cell area, we develop in vitro cardiovascular systems that allow us to do high throughput screening for the identification of therapeutic drugs. After having this expertise, we use our expertise in this knowledge. We use our expertise in advanced drug delivery systems to somehow deliver these drugs at specific cells, tissues in the body. So I'm also contributing for a called associative collaboratory in the center of region of Portugal. So this it's integrated from companies like in the IT sector, pharmaceutical, food industry, with the university as well, technology interface center, social providers as well, municipality, hospitals. So these stakeholders are integrating in this associated laboratory to tackle complex problems, and so in this case, aging. And so with this kind of setup, we hope that facilitate the translation between from fundamental knowledge to the clinic and to the market by facilitating the communication between all these entities. And we have also the support from the most important innovation cluster in Europe called IT Health. So this collab is in fact transforming the research, the health system in Portugal in two different areas. So active health, aging, as well multi-morbidities. And And in the next few years, we hope to enlarge these focus and these objectives to two more areas, two additional areas like palliative and left care, as well dementias, which are much more complex than these two first, but more differentiated, which much or a lot of should be done in order to solve these problems. So my talk will be split into topics. One is connected to stem cell derived models of cardiovascular aging, and the other is connected to advanced formulation, as I said in the very beginning. So for the first one, this avenue of research started 10 years ago or so, and was with our ambition somehow to create aged tissues in the lab that we could potentially use for studying aging biology, and also that with these kind of tissues, or we could somehow perform screenings to identify potentially anti-aging compounds. So in this case, we started to use inducible pluripotent stem cells as a model, rather than using like non-vertebrate or vertebrate models, in vivo models. And we start to use these because as a human signature, and so we could capitalize from that point of view. And because using these inducible pluripotent stem cells, we could differentiate into multiple type of cells that could be relevant for studying in more detail mechanisms of aging that otherwise would be difficult because of the rarity of the cells and because of difference in terms of metabolism between animals and humans. So, and for that we start to use accelerate aging model. So it's called Hutchinson-Gilford Progeria syndrome, typically recognized as progeria. And so I will speak about this in a few slides later. So in this syndrome, the patients accumulate a mutated protein called progeria. This mutated protein of lamina A, in fact, it's not only seen in Hutchinson-Gilford Progeria syndrome, but also in other diseases as well during physiological aging. And so we think that in fact, this syndrome can be inspiration for us to somehow study aging biology, as well to identify anti-aging drugs. So these individuals, they die quite premature. So the average lifespan is 14 years old. Nowadays, because of the treatment with one drug that has been approved by FDA 2 years ago, the lifespan is a little bit extended, but still very limited. And so there is for sure a need of treatments for these individuals. And so several groups, including the former NIH director, Francis Collins, they were very focused in studying the biology behind this syndrome, as well to identify potential treatments. So one of the most stronger phenotypes in this disease is this progressive loss of smooth muscle cells in large arterial vessels. And so that's one of the reasons why these patients, they die with the cardiovascular disease. In fact, so they die with stroke or myocardial infarction. And so for many years now, researchers have tried to identify the reason behind these loss. So this is a strong phenotype. It's probably the strongest phenotype in Progeria. But there are other phenotypes, including, for instance, like the accumulation of hyaluron and collagen in the blood vessels, as well vascular calcification and others. So in this disease, what happens is that so in wild type, so in non-disease state, so the protein is produced in a pre-form, right? So it needed to be processed, and there are a few steps that are identified in this slide until the end. There is a cleavage by a protease that makes these preform or these premature protein into a mature laminae. And so this is due to the cleavage by a protease of this farnesyl group that you can see here. In the syndrome, so in the AGPS, what happens is that there is a mutation in laminae, as you can see here. So you don't see the same kind of base in the genome. And so what happens, the protein is being produced that prevents the cleavage by the enzyme in the last step. And so there is the accumulation of lamine with this farnesyl group that is for some reason toxic. So lamine, for those that are not aware, is a lamine from the nuclear membrane, and so the accumulation of these progerin, which is the mutated form of lamine, induce different aberrations in the nuclei. So leading to nuclei dysmorphic nuclei, blabbing, and other aberrations in the nuclear membrane. So as you can see here, for instance, like the membrane is irregular because of the accumulation of the protein. So a few treatments have been developed in the last 10 years or so at the different levels: DNA, RNA, protein, cells, and some of them are identified in this slide. More recently, gene therapies and mostly gene editing therapies have been developed. For instance, like by Belmont as well, David Liu in Harvard, to correct the mutation. But still, what has been shown is only the treatment and potentially increase in lifespan, but only at preclinical models and when they are treated at early stage. So after these animals are born, if they are treated immediately by gene editing, they have a benefit in terms of lifespan. So it's still not clear if animals treated at a later stage, they can somehow be reversed. And of course, still there is no clinical trial ongoing. So we, inspired by this, we start a few projects some time ago where we differentiate AGPS, so EPS cells coming from these patients with AGPS, and we differentiate these into smooth muscle cells. As I said, smooth muscle cells is a very important cell, a key player in this disease. So these cells, they express at gene and protein level key markers of smooth muscle cells that are shown here. And very importantly, when these cells are undifferentiated, they do not express or very residual levels of protein, but when they are differentiating to smooth muscle cells, they start to show markers of progeria. So they accumulate progerin, and they start at the gene and protein level, and also they start to show dysmorphic nuclei as well, and blabbing in the nuclei. So one of the interesting facts in these cells is that they started to have a decoupling between cytoskeleton with the nucleoskeleton. So these cells, when we culture these cells in micropatterns, so substrate with pillars with the different dimensions, so with the 8 as well width, so this is in micrometers. So when we culture these cells, smooth muscle cells from a GPS or wild type, we do see difference, significant difference. So when these cells are cultured, the nuclei doesn't elongate, or in most of the micropatterns that we analyze, they don't elongate like a wild type. And this is because there is the decoupling between the cytoskeleton with the nucleoskeleton. So the nuclei doesn't receive information how they should behave in substrates with different topography. You know, so as you can see here, so the N is the wild type, so that's the known disease, smooth muscle cells. And in green is the AGPS smooth muscle cells. So the AGPS, they don't respond like the other cells, so in terms of elongation. In collaboration with Ali, we a few years ago, with the exchange of a PhD student, so João Ribas, he developed a very interesting microfluid system where he could take advantage of two different mechanical properties. So, using this microfluid system, it could culture smooth muscle cells in this PDMS membrane, and so we could perfuse the cells with medium. So recapitulating the flow shear stress, that these cells are exposed at some extent, even if they have on top endothelial cells, they are also exposed to this kind of flow shear stress. And also because of this two-chamber microfluid system deforming the membrane that the cells are cultured, it could mimic what happens in true blood vessels by the vasodilation of the blood vessels. So it could recapitulate two different, these two effects, physiological effects, culturing these cells in this microfluid system. And what you could see is that culturing these cells, you could see that there was, when he cultured with a cyclic strain, these smooth muscle cells at 16%, you could see an increase in the inflammatory cytokines, so EL6, EL1 beta, and also JAN. And also he could show that they have a kind of hypertensive genotype by the expression of CAV1. And so in fact, this was the first report showing that these cells can be induced in the inflammatory phenotype after cyclic or after vasodilation. And so nowadays, in fact, this is being explored as a potential treatment for the disease. Also, we develop a co-culture system, again, with a microfluid system where we co-culture smooth muscle cells with endothelial cells. And what we see is just capitulating or just capitalizing the flow shear stress. What we saw was very interesting fact. So when we culture, let's say fibroblasts with this disease, and we culture in flow under flow conditions in the microfluidic system, we could keep the cells alive, and so we don't have any loss of cells, even with this phenotype. So with the AGPS. Also, if we culture smooth muscle cells without AGPS, we could keep them alive in this microfluid system for 8 days or more, so without losing the cells. But when we culture these smooth muscle cells with endothelial cells under flow conditions with the AGPS, we start to lose the cells after a few days. And so again, this is very interesting because it's not just connected to the AGPS, because fibroblasts, we could not see the loss. Is really connected to the intersection of AGPS with the smooth muscle cell phenotype. And so this is very interesting. And we try to, making a long story short, we try to understand what was the reason behind this cell loss. And we found by RNA-seq and other kind of screening approach that one of the reasons, at least in our study, was that these cells secrete high levels of MP13, so a protease that is also very connected with aging. So again, there is kind of boundaries between this model and aging at all. So in fact, MP13 is a marker, is a marker of aging. And so this exists with AGPS smooth muscle cells, but not with other cells like wild type smooth muscle cells, or even fibroblasts cultured in flow conditions with the AGPS phenotype. And so we could show that inhibiting biochemically with the small drugs, small compounds, or genetically, we could keep the patency of these smooth muscle cells cultured under flow conditions. We did a transgenic mice where we cross a mice that has been reported some years ago by Carlos Lopez-Otin, that is the best model for Progeria, with another transgenic mice, and we were able to make different levels of expression of MP13 in these mice. And what we could see is that while mice expressing MP13 have a low level of cells, low number of smooth muscle cells in aortic arch, which is a large arterial vessel exposed to high flow shear stress. When we have the transgenic mice with low levels of MP13, we have increased patency of smooth muscle cells in this compartment, showing that the decrease in the expression of MP13 is important to increase the number of smooth muscle cells in these mice. And we think that the reason behind the loss of smooth muscle cells is because there is an increase of accumulation of heparan sulfate. So it's a component of the glycocalyx. Glycocalyx is a component of endothelial cells that is secreted. carbohydrates and proteoglycans that are secreted by endothelial cells, but also from smooth muscle cells. And what we believe is that the increased accumulation of the heparan sulfate in these cells after exposition to flow shear stress, because heparan sulfate acts as a mechanical sensor, we believe that it leads to the expression of MP13. So if we, in fact, if we remove heparan sulfate with an enzyme, in this case heparinase, we have a significant decrease of MP13 concentration, and we have also an increase in the patency, so increase of in the number of smooth muscle cells in the flow, in the microfluid system, showing that at least in part the smooth muscle cell loss when it's culture under flow shear conditions is due to the increase of accumulation of heparan sulfate. More recently, we identify formulations, nanoparticle formulations that are somehow eliminating more preferential HGPS endothelial cells. So in this case, I mean, we did a screening with the different kind of nanoparticles formulations, and we found some of them that are very with high tropism for endothelial cells with a GPS. So for instance, as you can see in here, so in gray is HGPS endothelial cells, and in blue, dark blue is the native APS cells, so without the disease. And for instance, for this formulation, this one is able to promote necrosis in endothelial cells with HGPS, while very minimal necrosis in native, so non-disease endothelial cells. So we hope these could potentially be an approach that could eliminate cells that accumulate high levels of HGPS in the blood vessels. So in short, as highlights, we have running protocols for the differentiation of these iPS cells into different cardiovascular cells. So we are key to collaborate if you see opportunities connected to these topics. We have, as I mentioned, in vitro models with these systems, with these cells, that could be a very interesting approach to study human aging. And although I didn't show, well, I show for the nanoparticles, but these systems can be very interesting also for drug screening. And we are presently doing some work on this. So in the second part of this talk, I'm going to speak about advanced drug delivery systems. It's very well connected also to Terasaki Institute or about scientific interest in this area. And so I will start by presenting some of the work that we are doing with two different kind of systems. So one connected to biological formulations. So in this case, extracellular vesicles. And in the second part, I will show you about synthetic formulations that most of them are activated by lights. So in the first part, I'm going to speak about the extracellular vesicles. So these vesicles, as you might know, are produced in cells. So they come from the invagination of the membrane during this trafficking process. So they accumulate components coming from the cytoplasm. So both the protein as well, non-coding RNAs, and then they are at some point exocytized, and then so they will retain part of the content of the membrane, and as well components that come from the cytoplasm. So they have typically above 100 nanometer, 150 nanometer in average. So these are data that coming from my lab for EVs coming from mononuclear cells from cord blood. And so these are all 10 image how they look like. So they kind of deform by the drying process that they are exposed, but they are so they form these kind of spheres when they are hydrated. So we in the last years we are trying to capitalize in the properties of these vesicles to as drug delivery systems. So although they have a cocktail of factors in the lumen, we want as a bioengineer to modulate the content, and so we are identifying components that somehow can facilitate the transaction of let's say small drugs or microRNAs into these vesicles. And so recently we did screening with a different approach, and we come up with a chemical approach that is very efficient to transact these vesicles with microRNAs. So using this approach, a chemical approach, so it's a kind of transaction agent, we could enrich very significantly these EVs with the microRNAs, and this is very compares very favorably to other approach like heat shock, detergents, or even microRNAs modified with cholesterol, as well electroporation. So this chemical approach is very interesting to enrich the EVs, and we could show that in fact these can be used for EVs coming from different sources. So in this case from mononuclear cells, EVs from mononuclear cells, EVs from urine, as well fetals, bovine serum. And this approach is very interesting because the enrichment that we have is much more significant that we have, let's say, if we transfect the precursor cell, so the EV secreting cell with the microRNA, and then isolate the EVs. So for instance, as a proof of concept, what we did was to transect mesenchymal stem cells with the microRNA of interest. And as you can see, the order of magnitude of enrichment, it's much inferior. It's much lower than what we have with our approach. So capitalizing. So in this case, what we do is we isolate the EVs, and then we transect EVs with the microRNA of interest. So it's pause isolation method. And so we are using these modify EVs, for instance, like for the treatment of, as a potential treatment of age-related disease. So in this case, Parkinson disease, in collaboration with a member, a close member of my institute. And what we show is that, so this Parkinson is being induced by the injection, in the intercerebral injection of this compound. And so this compound basically eliminates or kills dopaminergic neurons in the substantia nigra, as well in striatum. So as you can see here, the black, so the dopaminergic neurons are lost. But if we inject simultaneously the drug with these EVs enriched with microRNA 124. I'm not going get in details why we are using this microRNA, but it's a long story. It has protective roles in, and it's very important in the context of the brain. So if we deliver these microRNA when we induce the disease, as you can see here, it protects significantly the dopaminergic neurons, which is the most important neurons that are lost during Parkinson. And so this is not only at the cellular level that we can see this relevance, but also at behavior level. So when these mice are treated with these enriched EVs, they have less propensity to fall. I'm sorry. So they require much more time to fall than animals that are not treated. And also they don't have this contralateral net rotation, which is typical in Parkinson. So they have less propensity to have this kind of rotation. So we show that these EVs can have impact at functional level. Also, recently we use high throughput screening to identify microRNAs that are interesting for increase cell survival after transplantation. So as you might be aware, so these cell therapies, one of the gaps and limitations is the fact that they lose, so they die after a certain time. And so we start to think in developing to transfect these cells when they are transplanted with the EVs that could somehow carry a pro-survival microRNA. So we, by high throughput screening, so in my lab, we identify a couple of microRNAs that are shown here that are relevant for to increase the survival of these cells. And so we show that these EVs transfect with these microRNAs, we could show that they accelerate the healing process in diabetic mice as compared to EVs administered topically in these diabetic wounds with scrambled microRNA. So these EVs with these microRNA 425 5P was able to really accelerate the healing process. And what part of this is because it increased the survival of endothelial cells in these mice. As you know, endothelial cells in these diabetic mice is impaired. And so we believe that these EVs transfected with microRNA 425 5P increase the survival of these endothelial cells. We have also the possibility to track these EVs with the PET MRI imaging techniques. So we have very close facility, imaging facility, and so we have the capacity to label these with radioactive copper. So we can follow these EVs in mice. As you can see here, they tend to, in a short time, they tend to allocate in liver. Some of them secreted by urine, and some of them also accumulate in the kidney as well, in the lung, and very few in the brain. But because using this PET imaging platform, even if it's very low in the brain, we could have sensitivity to locate these EVs in the brain. And so this is very important if we wanted to monitor populations of EVs that accumulate at very, very low percentage in tissues and organs. Also, we develop drug delivery systems for the delivery of these EVs. So these, in this case, is a gel, either gel that is based in hyaluronic acid. So it's transparent, or as you can see here, it accumulates high levels of water inside. And so what we did was somehow to capitalize in Michael type addiction kind of reaction. So reacting these vinyl group with thiol groups in hyaluronic acid. And so these part of, we developed this linker. It's a cross-linking aging that has a photosensitive element. So if we shine a blue laser or UV, we can somehow cleave this linkage. So DVs will be chemically conjugated to the gel, to the polymer. So in black is the hyaluronic acid, and so if we shine the light, we can cleave this linker, releasing the extracellular vesicle. And so we show that using a specific protocol for activation with the blue light in mice, in this case with diabetes, we could show in a wound healing model, we could show that these mice treated with the gel containing the vesicles, if we shine the gel with this program of activation, we could release and accelerate the healing process in the wound. And so what we think with this is potentially this could be useful for patients with the chronic wounds having access to a laser at home, right? So the gel could be in the wound for several days, and potentially each day the patient could somehow stimulate the wound with these laser and potentially releasing the vesicle inside of the gel. Now I'm moving to the synthetic ones, so not more capitalizing the biologicals, but in synthetic. So this is one of the synthetic nanomaterials that we developed in the last year. So it capitalizes in a plasmonic carrier, so gold nanorods that have been modified with oligonucleotides. These oligonucleotides have a complementary sequence to an oligonucleotide that has been attached to a microRNA. So with this process, we can hybridize the microRNA to these oligonucleotide that has been attached to gold nanorods. And by playing with the hybridization temperature, with the melting temperature of this hybridization process, we could somehow, if we expose to an infrared light, so in this case at 780 nanometers, we could warm up because the gold nanorod will convert infrared radiation to temperature. We could warm up and deshybridize these microRNA and releasing it according to the activation with the infrared light. And so we were able to show that using this kind of strategy, we could potentially use to release multiple molecules, just playing with the difference in the temperature, the melting temperature. So In this case, we have a microRNA that is released at 51 degrees, while here we have a microRNA that is being released with a slightly higher temperature. And so doing this and using a reporter cell line that has promoters to specific microRNAs that are in here, we could see the knockdown of each microRNA. So if we release just using the same laser, so in this case we are using the same laser, so at 780 nanometers. If we release these microRNA or microRNA 155, we have the knockdown of M Sherry, because that's the protein that is being connected to the promoter of the microRNA 155. And if we release by increasing the power of the laser, so in this case is 1.25 watts per centimeter square. If we increase to 2 watts centimeter square, we can release a second microRNA. And so we have the knockdown of EGFP. So with this very simple approach, we can somehow orchestrate the release of molecules inside of the cells. This is another approach where we use, in this case, not inorganic, but organic. So in this case, we are using a polycation that has been modified with a photosensitive molecule that is complex to a polyanion. So these form nanoparticles. And so when these nanoparticles are exposed to a blue laser, they can be disassembled and releasing a small molecule. So in this case, we use retinoic acid as a proof of concept. So it's a hydrophobic molecule, so it's retained in the nanoparticle if there is no disassembly of the nanoformulation. And so if we disassemble, we can release the retinoic acid. And so this is a quite a fast process. So as you can see here, and very interesting, these formulations get inside of the cells and they accumulate in the cytoplasm, and they tend to accumulate for at least 6 days. And so this is very interesting because we can somehow take advantage of the transport properties of the cells, and then when they reach a specific organ, so in this case we are interested in bone marrow, if they reach to the bone marrow, then we can shine the blue laser and then disassemble and release the drug inside of the transporting cell. And so that's what we did. So basically what we did was to somehow transfect these kind of mononuclear cells that have tropism for the bone marrow. And so they were labeled with GFP. And so when we transfect these cells with these nanoparticles that are photosensitive to blue laser, so so initially these so these cells, they accumulate in the bone marrow, but so what we did was afterwards we use the same kind of cells, but now transfected with the nanoparticles, and so they tend to accumulate nearby in the proximity. And so if we shine now the laser in the target cells, they start to differentiate, and then they start also to crosstalk with the target cells, and they also initiate a differentiation program in these target cells. So these, we think that this could be an interesting approach to deliver somehow drugs at the place that typically are difficult to reach. And so we capitalize in these transporting cells to reach to compartments that otherwise would be difficult to reach. Also, we did a bunch of formulations, libraries of these formulations, and we are keen to collaborate also in this context. So we have formulations that respond to light with the different physical chemical properties. So if you are interested, let me know, and I will be definitely interested to collaborate. So these are very interesting to transect cells, and so if we compare, for instance, with lipofectamine, when we activate these cells that are identified here, they have much more transfection efficiency. So here we use a short interference RNA that knockdowns GFP, and so we have much more efficiency than lipofectamine. And I will finish my presentation just speaking about the gene editing system that we developed very recently, that is connected to a Cree recombinase system. We have also running a CRISPR-Cas9 system. So it capitalizes in the delivery of the protein, not plasmid. We use synthetic formulation, not a virus, and using this system we have spatial control, and afterwards we can control, or we can evaluate the activity of the gene edited cell using optogenetics. So this is the system where we mobilize the Cree recombinase. It's capitalized in a linker that is again activatable by light, and it has a component that is very important somehow to promote the endolysosmal escape. So this hydroxychloroquine prevents the fusion of endosome to lysosome, and then facilitates the crossing of the protein to the cytoplasm, and afterwards the protein can reach to the nuclei where does the recombination process. So again, so in this case, it's promoted by near-infrared. So this is an up conversion formulation. So it converts near-infrared light into a blue light, and so it's the blue light that is able to cleave this linker and release the Cree recombinase. So it's a very fast process, as you can see here. So the release of the protein after the stimulation with the near infrared. So this is activated at 980 nanometers, as you can see in here. So this is a very fast process. So we use a reporter line where we can measure these in a very quantitative way. And after 24 hours, almost 100% of the cells are recombined with this approach. And this is very efficient compared to other transfection systems developed and reported in the literature. And very importantly, we have a single cell control. So we can, so using multiphoton, we can, so in here we have two, but because the single cell divide, and so you can see here too, but we have resolution at single cell using this kind of system. So these we can use to transfect and to do the recombination in neurons. So this is in neurons. We can inject the formulation in the brain of transgenic mice that is sensitive to Cree recombinase. And in the contrast to the virus, the nanoparticles accumulate in very specific regions in the brain, in the regions that we deliver. While with the AAVs, they tend to migrate and go far away from the regions where they are injected. And we could see by combining this editing formulation with optogenetics that we could somehow show that the dose that we deliver, so in this case we delivered the editing formulations in the VTA, which is a kind of rewarding kind of region in the brain. So what we did was to edit. So basically these neurons will have channel rhodopsin. So for those that are not aware of the optogenetics, is the protein that is sensitive to light, to blue light. And so this region of the brain will have this protein, channel rhodopsin, because of the ability of the formulation to do the addition. And what we did was we allowed to express for a couple of weeks, and then we did a very simple experiment, a behavioral experiment. So if the mice, so we allow mice to choose one part of the region of this cage, so in the black or in the white part, and so by default they will select one part of the cage. And so what we did was in the part of the cage that didn't select, we activate with the near-infrared light. With the blue laser. With the blue laser. And so basically, what happens is that this part of reward region of the brain will be ions will cross and will do the depolarization of these neurons. And so basically, so the animal will feel like a reward. And so afterwards, we wait a couple of more days, and then after the reward, we did again the test and see if the animal will select by default the same region of the cage. And because we did in the part of the cage that was not the primer or the preference for the animal, now the animal select the one that was not selected because of these kind of reward trigger in the brain. So this is a simple demonstration that we could edit at the levels that we could see at functional level the effect of the editing. Okay, so I will finish. And so basically we have biological synthetic delivery systems that are can be useful for the delivery of small molecules, non-coding RNAs, as well messenger RNA and gene editing formulations. We have also systems to monitor these nano formulations and drug systems, hydrogels, to release them. And we again with this last example, I think we demonstrated that these systems can be very useful for gene editing and for to somehow correct mutations or do epigenetic reprogramming. Thank you so much for your attention, and again to thank the invitation from Ali Kodamasani as well, Mehmet, for giving this talk. Thank you so much.
Thank you for the fantastic talk, Professor Lino. Several questions came in. What is the correlation of heparin sulfate molecular mechanism and progeria?
Exactly. So heparin sulfate is it accumulates in blood vessels, so in the large arterial vessels in the progeria. So it's in fact one of the hallmarks of the disease. So heparin sulfate accumulates at high levels. So it's in fact in biopsies from these patients is that there are some documents showing that it accumulates quite significantly, and so we and from previous studies, not in the context of Progeria, but in others studies, it has been shown that heparan sulfate, it's a kind of mechanical sensor. So somehow it triggers or it senses the flow shear stress. And we believe that the high levels of accumulation of this heparan sulfate somehow makes more sensitive these vascular cells to flow shear stress. And so we believe that is one of the reasons why smooth muscle cells are lost from the vessels, even more than endothelial cells.
Thank you. Is there any molecular level correlation of progeria and Alzheimer's disease progression?
That's a very interesting question, Mehmet. I don't know. So it's very interesting. I don't know even if someone explore or investigate that connection. So yeah, that's what I can say.
Thank you. Is Kevlin lipid raft pathway connected to endolysosomal pathway and further exosomal exotisis pathway?
So again, sorry, I didn't understand the first part of your question.
The Kevlin lipid raft pathway, is that connected to the endolysosomal pathway and further exosomal exotitis pathway?
Yes, so it's connected to endolysosomal pathway, but so it's unclear which one. Still, I mean, there are not so much yet knowledge which one is the most efficient somehow to lead to the endosomal escape or to exocytose. Okay, so for instance, like in the context of biological barriers, for instance, like in the context of BBB, right? It's still debatable which one is the one that is the most efficient to allow the transcytose. So meaning, so if a formulation that gets inside of the cell by caveoli, if is the one that is more efficient, that leads to transcytose or other pathways. So it's still debatable in the scientific community which one is the most efficient.
Thank you.
I don't know if I addressed your question.
Yes, thank you. Yeah. What is the method for synthesis of EVs and microRNA?
Say again, sorry?
What is the method for synthesis or EV microRNA?
Okay. So these EVs, they are somehow, so basically there is a invagination of the membrane. So there is kind of molecules that I'm not disclosing here that leads to this kind of endovagination. Okay, so this is a intracellular process regulated by some molecules. Okay, and doing this in the multivesicular body, which is the MVB, rather than going to the lysosome and be kind of degrade. What happens is, again, it's a kind of signaling process, intracellular signaling process that I'm not disclosing all the components, molecular components. So this leads to diffusion to the cell membrane and then the release of these vesicles. So these vesicles, in fact, they contain components that are coming from the cytoplasm of the cell. And so if you, let's say, transfect the cells with let's say, small drugs or microRNAs or whatsoever, they some of them at least tend to be accumulating in these vesicles. So people believe that these vesicles are a kind of cell-cell communication at a long distance. So they are involved not only in homeostasis, but also in pathology. So so there is work showing, for instance, like in cancer cells, part of these kind of formation of metastasis at the long distance is mediated by the secretion of these vesicles from the primary tumor that are secreted, and then they tend to accumulate in specific regions of the body that somehow prepare somehow these kind of metastasis sites where cancer cells afterwards tend to accumulate. So this is not only for beneficial or in terms of kind of communication in healthy systems, but also they also are involved in pathological communication process.
Thank you. What is the route of administration of your light-induced hydrogel? Topical route, or is it topical, exactly, or is it subcutaneous?
Exactly, it's a topical. So what we do is injecting. So in here, so in here we use a topical administration. So the wounds were somehow induced in the back of the animal. So we inject as a liquid. So they gelify in the wound. Okay, so when we inject, they are liquid. So they gelify in the wound, and then after gelify, we started to degrade the linkers by using these blue lights trigger. So that's how we do, so that's topical administration. But potentially this can be used for laparoscopic administration, for instance, like in other settings. So this could be relevant, for instance, like for myocardial injection or even in other contexts. Yeah.
Thank you. Is there any similar expression of specific microRNAs in Progeria, Parkinson, Alzheimer disease, or other brain diseases?
Yeah, so there are a few microRNAs that are kind of shared by some pathologies. Yeah, but it's still a lot of research being done, to clarify the role of each one and which kind of cell they play. So it depends in multiple factors, I will say. So in our case, we are using for regenerative or potentially as an anti-aging drugs, and so it depends in the cell type. That's what I can say. Yeah. So it depends in the context.
Thank you. Maybe chloroquine is used for malaria. Maybe any interaction with spleen and RBCs. In place of chloroquine, is it possible to use other safe molecule for endosomal escape?
Yeah, definitely. I mean, in our hands, hydroxychloroquine was very beneficial because it somehow, the mechanism is still not fully understood. But in our hands, what we found was that these hydroxychloroquine somehow prevents the fusion between, let's say, early endosome with the lysosome. Okay, so preventing these somehow gives more space for the the content that is in the endosome to escape. Okay, but there are others. So in fact, people, some prior art showing other strategies. For instance, a couple of years, people use kind of modify peptides that could somehow prefer the endosomal membrane. So that's one strategy. Also, some years ago, a paper published in Cell, they explore a kind of surfactant that was able to perforate the endosomal membrane. Yeah, so there are a couple, but in our hands, when we compare, so in fact we compare because some of these approach, they use a recombinase readout to show the possibility to deliver proteins. So we could compare side by side our strategy with the strategies that have been reported in literature, and side by side we could show that in fact our system was very efficient. In some case, tenfold more efficient than these systems, the state-of-the-art systems for cell transfection with the protein. So we were very happy to see that. Yeah.
Thank you. Well, that's all the questions I have. Thank you for your time, Professor Lino.
Thank you. Thank you so much.
Yeah, have a nice day.
Yeah, nice to see you all. Thank
Same
you.
here. Visit us. Thank you.
Bye-bye.